A little over a week ago, new research was published about the sequencing and assembling of the hookworm genome in the journal Nature Genetics. Joining me on the phone is the senior author of the paper, Dr. Makedonka Mitreva, PhD. She is an assistant professor of medicine and of genetics and a member of the Genome Institute at the Washington University School of Medicine. Hello, Dr. Mitreva, and thanks for joining me today.
Hello. It's nice to be with you today.
Thank you. Okay, before we get into the research, can you please tell my radio and website audience, what is the hookworm and what kind of pathology does this cause, what kind of disease does it cause in humans?
Hookworm is an intestinal parasite, is a roundworm with a body size of an adult hookworm being only 1 centimeter in length, and the parasite lives in the small intestine of humans. The adult worm attach to the intestinal mucosa where they feed on blood, and each adult hookworm sucks up to 30 microliters blood per day and can survive in the host for up to a decade. A female worm can produce up to 10,000 eggs per day, and the hookworm infection is characterized clinically by intestinal blood loss, resulting in anemia, malnutrition, especially in pregnant women, and a stunted growth and learning problems in children.
Okay, and how does one contract hookworm?
The hookworm life cycle begins when the adult male and females mate in the small intestine of humans, and the female produces eggs that are passed in the feces. Those eggs can then contaminate the soil, and in warm and humid conditions, they will hatch and develop to the stage where they can become infective. This infective stage then infects humans by penetrating through the skin, for example, through bare feet, and it migrates through the blood system to the lungs. And then actually you get a cough, you cough them up into your mouth, and then you swallow them, and they go down to your intestine. Skin penetration is the infective stage, and is the only infective stage for the species that we worked on. And that species is responsible for 80% of the infections. Because it transmits through the soil, the hookworm is endemic in countries with lack of sanitation. So the other hookworm species that infects human transmits by ingestion of the infective stage, and then the life cycle is shorter because it doesn't go through the lung, but it settles directly to your intestine. This process from infection until parasitism establishment takes about 5 to 9 weeks.
Very good. And how is hookworm treated, and is there a preventive vaccine for it?
So hookworm infects approximately 700 million people, mainly in disadvantaged communities in tropical and subtropical regions. And presently, the treatment of hookworm disease in these endemic regions relies mainly on mass treatment with a broad-spectrum drug called albendazole. While it is an effective therapeutic, this drug does not prevent reinfection, which frequently occurs since this parasite is transmitted through the soil. Because of the reinfection, there is a need for repeated and extensive use of the drug, which has potential to lead to treatment and drug resistance, which has actually been reported recently. Anti-hookworm vaccine is urgently needed in countries that have high rates of hookworm infections, and there is a determination from some philanthropic organizations to facilitate development of a vaccine, and such efforts have been funded, but as of today there is no commercially available vaccine. And this progress in developing novel preventive and control strategies has been hampered by the lack of understanding of the biology and the infective process of the hookworms.
Okay. And you mentioned that there's, at any one time, about 700 million hookworm cases throughout the world. So it's a huge public health problem. How common is the parasite in the United States?
Um, so, as I mentioned before, hookworm infections are common in underprivileged communities in tropical and subtropical regions, mainly in areas of extreme poverty where the sanitation infrastructure is underdeveloped or it does not exist at all. There are reports that in early 1900s, like 1910, for example, an estimated 40% of the population of southern United States was infected with hookworm. But the number of infections were dramatically reduced through mapping the disease, which means identifying the exact states where the disease was a problem, massive drug treatment to cure patients, and providing education, which included education for proper hygiene and lifestyle, such as the importance of wearing shoes, for example. As a result of such an action, the disease was reduced and eventually eradicated, and today it's not a public health problem in the United States. Most hookworm endemic regions are in Africa, Asia, and South America.
Okay, great. Well, let's go to your research. You unraveled the genome for the New World hookworm, Necator americanus, which is the most common species, as you mentioned. Without getting too technical, what did you learn from decoding the genome?
First, if one compares the size of the hookworm genome to the size of the human genome, the hookworm genome is only 8% of the size of the human genome. There are two main reasons for that. It is a less complex organism, of course, and a second one is a parasite, which means it relies on the host, so many processes do not occur in the parasite. However, the number of genes each of them encodes is much more similar. Our approach enabled understanding of different aspects of how the hookworm evades the body, how it feeds on blood, and how it evades the host immune system. We know exactly what are the genes that are expressed in different stages of the life cycle. So by decoding the hookworm genome allowed us to discover the combination of genes that orchestrate each of these processes and to identify specific targets that might be vulnerable to vaccine or new drug treatments. By decoding the genome, we also showed that hookworm has undergone a considerable and unique expansion of molecules that the parasite uses to evade the host immune system, which allows the parasites to survive in the host for a long period of time. So, in addition of identifying some such a vaccine and drug targets, we also undertook an advanced approach to prioritize those targets so that scientists can quickly follow up on the ones that appear to be most promising.
Okay, so my last question is, what key medical advances from knowing the genome do you suspect will be used in the future?
I can see several medical advances coming from using the genome in the future. First, regarding diagnostics of hookworm infections. We identified hookworm molecules with low similarity to proteins in other species, but recognized by all the infected individuals we tested in our studies. And those molecules demonstrate extremely high diagnostic potential. Second, regarding novel anti-hookworm therapeutics. We now better understand the hookworm infectious process, the blood feeding, the survival within the host, and we use that knowledge to identify molecules and processes that are essential for survival of the parasite, making them a very good target for developing therapeutics. And third, what I did not mention so far is the growing interest of exploring hookworms and other roundworms as treatment against non-infectious diseases in industrialized countries. So hookworm suppresses inflammation in the intestine to protect itself from detection of the host immune system. So you can imagine that you will be a successful parasite if you can make the environment you are in as healthy as you can. So such a mechanism that hookworms use to do may prove valuable to treatment of non-infectious diseases such as autoimmune diseases and allergies. In fact, hookworm products have been used in clinical trials against such diseases. So it is our hope that the new research can be used as a springboard not just to control hookworm infections, but also to identify anti-inflammatory molecules that have the potential to advance new therapies for autoimmune and allergic diseases.
Very good. That's very exciting. Well, your research, your paper was very interesting. I want to thank Dr. Makedonka Mitreva with the Washington University School of Medicine. Thank you, ma'am, for your expertise, and have a great day.
Thank you very much.
You're welcome.
Bye-bye.